Hi everyone, welcome to my presentation on circular RNA. Some RNAs in your body don't make proteins. These are called non-coding RNAs or ncRNAs. There are a lot of types of ncRNAs like siRNAs, tRNAs, and miRNAs. In this video, we'll talk about a special type called circular RNAs or CircRNAs. CircRNAs are like regular RNA, but instead of being a straight strand as shown here, they form a circle. Imagine a piece of string that you tie the ends together. It's kind of like that. This circular shape happens through a process called backsplicing. In backsplicing, the beginning and end of the RNA link together to make a loop. What's cool about circRNAs is that their circular shape makes them tougher. They don't break down as easily as regular RNA because there are no free ends for the enzyme to grab a hold of. These RNAs come from different parts of the genome and can be made from exons, introns, or a mix of both. To understand circRNAs, we need to know how regular RNA is made first. This process is called canonical splicing. It's like editing a video. You cut out the boring parts, the introns, and keep the interesting parts, the exons. Here's how it works. The spliceosome, a tiny machine in your cells, finds the boundaries between introns and exons. It cuts out the introns, forming intermediate loops called lariats. These are thrown away. At the same time, it glues the exons together to make a complete message called mRNA. The removal of intron and the rejoining of exons happens at the same time. This mRNA is what cells use to make proteins. But how are circRNAs formed? CircRNAs are made through a special process called backsplicing. It's a remix of regular splicing, but instead of joining pieces in a straight line, it links them into a circle. There are two main ways this can happen. The first is by using repeated sequences. Think of these as matching puzzle pieces around the circRNA. They help the ends find each other and link up. The other is with helper proteins, depicted here as the blue ovals with RBP. These proteins act like glue to bring the ends together. Interestingly, the parts of the RNA near the circle are longer than usual and have special repeats that help this process happen. The result is a loop of exons or a loop of exons plus intron. But why are circRNAs important? These molecules have some important jobs in the cell. One of them is gene regulation. CircRNAs act as sponges for microRNAs or miRNAs. miRNAs usually stop certain genes from being active, but circRNAs soak them up so they can't do that. They're also involved in regulation of transcription and alternative splicing, as well as cell communication. CircRNAs can be sent out of cells in tiny packages called exosomes. These packages carry messages between cells. They're also involved in building platforms. CircRNAs help bring proteins together to do specific jobs. Think of them as scaffolding on a construction site. CircRNAs can also be used as template for translation and are involved in immune response. CircRNAs are especially common in brain cells and tend to build up as we age. Scientists are still studying them, but they know circRNAs are linked to diseases like cancer. There's still a lot to learn about these fascinating molecules. Here are the references I used for this project. Thank you all for listening.